Hey, I'm Ryan,
And I'm Corey.
and this is our biology project for the stages of mitosis. So first we want to get started with the first stage, which is interphase. We're also first hour biology. So first, as you can see in the first picture, the chromosomes are duplicating, and pretty much after that the copies remain attached to each other. Now, it is not technically considered a phase of mitosis as well. And I mean, in general, this is that one stage where the cells are just doing everyday activities, and the cells look like everyday cells with a dark nucleus. And the goals of mitosis, this is also the stage where they're prepping for mitosis as well. So the one cell will become two, and to produce an exact copy of the original cell, which is natural cloning. Replication occurs during interphase. This is why it's not considered a stage of mitosis, pretty much, and has twice the genetic information than normal. But then, as you can see in the second picture, this is what happens when the chromosomes duplicate.
The second stage of mitosis, or technically the first stage, is prophase. And in prophase, the nuclear envelope begins to break down, and chromosomes form. They shorten from DNA and wrap around histones. And in the nucleus, chromosomes condense and become visible. And in the cytoplasm, the spindles form around, and as the envelope begins to break down.
Alright guys, so our next stage of mitosis is metaphase. So firstly, the chromosomes are moved by the spindle fibers to the middle of the cell. After that, all the chromosomes line up pretty much, like I said, in the middle of the cell. The copy chromosomes align in the middle of the spindle as well.
Alright, the next stage of mitosis is anaphase. And in anaphase, the spindle fibers shorten and they pull the chromatinds apart. Chromatinds are pulled towards the poles north and south, and the chromosomes separate into two genetically identical groups and move to opposite ends of the spindle. There is a gap in the middle of the chromatinds that are pulled away from each other.
Alright guys, our final stage is telophase. So firstly, the chromatids, now called chromosomes, are at opposite poles of the cell. This is also when the nuclear envelope will reappear. As you guys saw from our last stages, the nuclear envelope did disappear, but this stage it does reappear back. Also, each pole should be a replica of the other side, and also, of course, the nuclear envelope reforms. The nuclear membranes form around each of the two sets of chromosomes. The chromosomes begin to spread out, and the spindle begins to break down, breaking apart cytokinesis, which is what cytokinesis means, pretty much breaking apart, pinching off a cell, and also the daughter cells pop apart. Not considered a stage of mitosis. Cytokinesis is not considered a stage of mitosis. The cell splits into two daughter cells, each with the same number of chromosomes as the parent. In humans, such as cells have two copies of 23 chromosomes and are called diploid.
Alright, thank you for listening to this project, and we look forward to making more.